Hey, good evening, everyone. Welcome to the today's webinar of how to plan a research project worth publishing. I am Dr. Prasanna Kumar, and I profusely thank Barcode Biotechnology for inviting me as a resource person for this webinar. Okay, so I want to deliver this message in the first slide itself. What are you seeing here in the right hand side is actually a template reply from the European universities and Canada or US universities. So when you apply for a master's or PhD, this is what they will reply. No, most people will get this reply. Your application has been carefully considered by academic selectors who was impressed by blah blah blah, but you did not match the required criteria. What is this required criteria? Do you have a publication? Thousands and thousands of people will be having a MS degree or even a PhD degree. When you want to go for a higher studies, your skill is reflected by not by your degree but by the publication. The specific authors who want to select you for your higher studies or anything will be depend on what kind of publication you have produced. Okay, if you did not publish, you will perish in academic section. That's the golden rule. You will see this. I realized this after 30, but you will see when you are 21 or 23, that's a time you need to think about publication. What is a research publication? We will see. So far you have read a textbook, which is a consolidated material accumulated from the research body. That is research journals. From there, they have consolidated the material. They have given you as a textbook. Most probably you are reading something at least 15 years old. Okay, all those factuals and other things are many, many years old. Okay, that's not a current data what you are reading in the textbook. And you will see it is with time, even fact. Many facts have changed. Okay, so let me talk about how to select a valid research question. When you answer that, which will become a publication and it will get high citations. Okay. Let me start from very basic human thought process. Okay. See, whenever you think you have a new idea, most probably it is already a proven fact which you may not realize. After you produce your data by doing an experiment, you will come to know that this fact has been proven long back. To avoid this, 99% of master students and PhD scholars see master thesis where they are right now. All your senior master theses are in the library sleeping. You know, they will not find an expression as a publication. Why? Because they did not consider what is already done in the field, meaning they did not do a literature review. This is something I feel it's very much lacking in Indian ecosystem, where reading and understanding a literature, research literature is very much important if you want to contribute something meaningful to the research. You may think whatever you want. Those are just permutation and combination of thought process or impressions you have accumulated. Most probably, more than 99.9% time, it is already done, or it is not worth doing. They will be classified in these two category if you don't read the research article. Don't forget, we are the people who thought every planet is revolving around this earth, not the sun, once upon a time. Okay, so acknowledging the body of knowledge existing in a particular domain is very much important before you start any research work. So how to plan a research project worth publishing? This is my strategy. Okay, definitely you will find expression and you'll gain more citation if you follow this strategy. All right. First of all, select your domain. You want to do a research. Doesn't matter what kind of science domain or whatever is your domain. Select a particular domain that you want to do a research. Understand the domain. Acknowledge the existing knowledge. Then formulate a research gap. Without knowing what is the research happening in the domain, you cannot formulate a research gap. Then converting the research gap into research question. To test the research question, design a hypothesis, derive a protocol, and then start testing your hypothesis in forms of experiments. What I'm going to do in next 30-40 minutes is I'm going to explain you with an example how this will work. Okay, this is a simple strategy. The same slide I will share at the end of this presentation. See, I see for UG students or PG students, these are the three choices. Okay, either you will go for a higher study, go for a job, or you can start your own startup. I would say for all these three options you have, you need to know how to read and understand a research article. Without that, you are not going to progress in any of this selected domain. Ask any CEOs of startups, in Bangalore, Hyderabad, or New Delhi, the life science startups, they're all born out of research article. Once you cannot understand research article, you will never know how to do a life science business. Even for the job, join a biocon or any other leading life science industry, first day, first thing, maybe for 3 months, you will be asked to read the research article in the current domain, understand what's happening. For PhD, there is PhD or MSc or even postdoc. There is no way you can select a research question unless you read a research article. So reading, understanding research article is the first step towards publishing one. All right. See, I don't want to get into a theoretical part. Okay, basic research, applied research, whatever is your interest, you can choose one. But you understand that from here on, this presentation is not just a presentation or exchange of information. I want you to pause the video whenever you want and work with me so that at the end of this presentation, you'll have a research question. I can guarantee you that. So if you want to pause the video, you can pause wherever you want. From next slide onwards, I will tell you what are the common mistakes and how this all starts. So before progressing to next slide, I want you to do a small homework. Okay, pause the slide here. Try to formulate a research question or a research project in your mind. This is the area I want to work. Write down why you like to work in this area. What is the problem? How are you going to provide a solution? Write down what is the background of the work. What is the problem? How are you going to provide the solution? Maybe 3 lines, 5 lines, that's okay. Writing down will, clarify your mind. That's why I'm insisting again. Write down, then you can, uh, play the video and carry on. See, I told the same to a student. So the student wrote, isolating a bacteria from human skin, identifying new strains, finding out strain is harmful or commensal. I don't know why the student left can be beneficial. The student have no idea. The strain, it's a microbiology query about skin microbiome, microorganisms living in skin. Okay. If the identified bacteria is a harmful, what she want to do with antibiotics, so the student narrated, and then comparative study and other stuff they've been talking about. Okay, so this is a hallucinatory or imagination, most probably for master student or even PhD scholar. Before they initiate your work, this is a hallucination you have. Just pen down your idea. That's a step number one. Now step number two, copy and paste it in Google Scholar. Let's see what kind of you know articles you will get. See, the first result is cited by 1094 times. The skin microbiota source of disease or defense. If I read that paper, it can clearly say what are the common bacteria will end up isolating and other stuff. Okay. Yeah, I again went scroll down. I saw another paper, human skin microbiome impact of intrinsic and extrinsic factors. So I thought, okay, I will get into this paper, and this is specifically about skin microbiota. Let's start reading about it. If you start reading about it, you will know the body of knowledge already present in human skin microbiome. See what kind of bacteria phyla will be dominating, what will be the concentration, even the anaerobic bacteria, how they are found inside sebaceous gland, what are the common bacteria to isolate, not only bacteria, what kind of yeast, other fungi, even viruses, how much are culturable, how much are not among microbiome, every information you can get. Even you come to know the shiningness of the skin is actually because of lipoic acid secreted by Staphylococcus epidermidis. So immediately you got an idea why this epidermidis, if it is secreting this, why can't I use this bacteria to increase the shininess of my skin? Okay, but instead of just going for another Google search, I just went to Amazon. It is already there. The products are already there. Not only one, multiple products are there where they have using lactobacillus plantarum and other probiotics to increase the glow of the skin. Okay. Then I kept on reading the paper. What is the density of bacteria reside, and what are the other, factors influencing the microbiome? There are body of knowledge, from which body part, what kind of bacterial phyla can be, detected. Front foot at the heel, dorsal ventral of your hands. What is the bacterial phyla frequently there? Even with due time of one's human life, even based on gender, teenage, adult age, old age. These are the color represents, and the purple represents firmicutes, actinobacteria are represented by yellow color, proteobacteria by green color, bacteroids by blue color. And see, the purple, the color represents the major bacterial microbial phyla you will find in different age groups. Even same mother giving birth to two different babies, one through natural, other through cesarean, will influence the baby skin microbiome. The data is already there. Likewise, how your textile clothing is influencing the microbiome, how you intake the antibiotic intake you are exposed to actually can influence your skin microbiome. Many work has been done. Imagine the first imagination I had, that I want to work with, thinking that all bacteria are actually causing disease. It's completely wrong. Once you started gaining knowledge in your selected domain, you will find how ignorant you are. This is the first realization should happen before you start your research. Now, come to step number 2, how to find a research question. Now I have realized I'm so stupid to consider that skin microbiome have a commensal and only pathogens. I don't even know that actually the microbiomes are the one who keep the skin healthy. Now I know that I want to do research in skin microbiome. Sorry. So I will go to step number 2 now. There is something called Google Scholar. Here I am using my domain name, skin microbiome. Okay. So here at the left hand side, I can see anytime, and this search is done 2 years back. So now you can see since 2025, select the current year and read the paper. When I select the current year, I came across a review article, an update on current understanding. Why current? Why need to select the current year? See, we are going to find a research gap, the unanswered question. Better it is not from 1998, which might have answered on 1999. Okay. There is no meaning in spending time to understand a research question from the old research article. It's very much important you find a current year. Okay. So those who are actually into current year, those who already know this, may face difficulties in downloading the full text. It's not always true. Many quality journals actually publish open access, meaning you can download the full text, or you can use Sci-Hub, or there are many Facebook groups where you can post your DOI and get your PDF copies. There is a research platform called ResearchGate where you can actually find the authors. Authors have a rights to share their own work for personal request. Okay, you can request and get the full PDF. So there are many ways to access it. So since I am finding a current research gap, I'm started reading this article. Okay, current understanding of skin microbiota. You should know the difference between an English essay and a current scientific writing is this. See, these numbers indicates a citation. To write a one paragraph, this author has used 17 citations. Each and every line should have a proof of from where it is taken. It cannot be a story. Even if you say E. coli is gram-negative bacteria, you don't have to, you have to show a citation. Even for common facts like this, sometimes citations are neglected, but each and every line in a research article should be cited or should be provided with the proof. It should not be a imagination or hallucination. That's what it meant. So I kept on reading. I came to know unmet research questions. There is a separate topic. I'm very glad to see this heading. So the efficient protocol for extracting high quality DNA for metagenomic studies sorry, missing from the skin microbiome research. It's understandable because microorganism, when compared to artificially grown in the lab, the concentration is significantly high on the surface of the agar when compared to the surface of your skin. So DNA extraction protocols are missing. Many are studying about bacteriobiome, what about archaeobiome, microbiome, virobiome is missing, and how these communities are interacting is missing. How to distinguish between microbiome dependent and independent skin diseases is missing. So when I read through the article, you can also download this article and read through it. When I read through the article, these are the various gaps I'm seeing. Okay, various research questions needs answering. So now you see how was my imagination initially thinking that I'm going to isolate a harmful commensal, blah, blah, blah. But after reading a research article, I have a set of questions which is unanswered. It is from current year's research article, definitely unanswered. So should we always select a review article for understand a research gap? No. You can still select a research article with the data and other stuff. Usually in the discussion part, authors will be, writing about what was the question, how I answered it, what is yet to be answered. If you understand what was the question and how he answered it, yet to be answered is your research question. You can simply take and start working on that. Okay. So far we have seen how to pen down an idea and select a core domain. From that, Google Scholar current research article, how to take a research question. This is how the research should be progressing. If you can understand this, say for example, the first question, development of efficient protocol, I'm not going to just believe that the author says that it is unsolved. Again, I will go into Google Scholar. I have found a similar article, efficient model for high bacterial extraction, but there are a lot of lacunae and a lot of things needs improvement. So we cannot simply accept this protocol as a PACA protocol for extracting the high quality DNA from the skin. So from here, definitely such a protocols is an essential for the hour. If you can select through your master research, if you can select this protocol, if you can design a protocol or design a chemical reagent that can yield more DNA from the skin microbiome, you have a you have answered the unmet research question. Now if you publish this article, definitely you are going to get hundreds of citations. Okay, so that's how to that's how we should know what is required in the society, and to answer that, to fulfill that, we do research. Research should not be started with your hallucination and imagination. No one has done it, so I want to do. No one has done it because it is not worth doing. That's why no one has done it. Remember this always. All right. So we have selected a domain. We have asked for a research question. So what is this hypothesis? Why to have a hypothesis? To test a research question, hypothesis act as a bridge between your research question and the experimental workflow. The list of experiment you are going to do to achieve the or answer the research question. Understand this, single research question may have multiple hypotheses. Every hypothesis may have their own set of workflow. Unless until you answer your research question, you can modify your hypothesis and create your own workflow. Let me answer. What is this hypothesis? So let's consider this workflow. Okay, you have identified, you have formulated a hypothesis. Hypothesis can be of two types, null hypothesis and alternate. What is this null hypothesis? Does drinking tea reduces the risk of heart development? This is a question if you want to answer. The null hypothesis will be drinking tea does not reduce the risk of developing heart disease, whereas the alternate to the null is the alternate hypothesis, which says that it reduces the risk of heart disease. So to check the hypothesis now, if I want to check null hypothesis, I can develop my own workflow. If I want to check the alternative hypothesis, now I can develop different workflow. Okay, so hypothesis is a bridge between your research question and the experimental workflow. All right. You see, I will give this example. A author has declared, this is a famous paper which declared that a global oceanic nitrification rate is going to go down because of ocean acidification. Everyone knows that we are continuously burning the fossil fuel, releasing carbon dioxide, which is absorbed by ocean as carbonic acid. That decreases the ocean pH. So this author has made a claim that the ocean acidification is going to reduce the nitrification rate. So an alternative hypothesis was framed because there are other organisms like archaea, a prokaryote, that can take oceanic urea. Urea is not only from land runoff, Many organisms are releasing urea during excretion that can be used by archaea, which is not at all affected by acidification. Because urea is an inert molecule on cleavage, they can produce two molecular of ammonia inside the cell. This cleavage and enzymatic activity can happen inside the cell, thereby releasing ammonia, which can also undergo nitrification. So this is an alternative hypothesis. If archaea can degrade urea, then nitrification rate will not decline. To test this, this is actually my hypothesis. We formulated, I got a funding from Chinese government long back. I have published this in a research gate also. You can check this proposal. We successfully proved that acidification is not so. They may not decrease because of acidification, because only this author has considered only bacterial group, not the archaeal group when calculating these parameters. You can check this proposal. Okay. So hypothesis act as a bridge between your research question and your experimental workflow. Or other way of putting is your experimental workflow depends on hypothesis, which depends on your research question. So let's see. Once you have a hypothesis, you need to have a workflow. How to design this workflow? Like I said, unless until you read multiple papers, you are not going to understand what is happening in the research, how the workflows are designed, and it is very difficult for you to design the experimental workflow, for which I would suggest literature review matrix should be adopted. What is this literature review matrix? Anyway, you are going to read multiple literatures, right? So once you have a hypothesis, whatever literature that aligns very close with your hypothesis, you are supposed to read, cite the author name, the title of the paper, short title, what is that hypothesis they have tested, what is the methodology they used, what is the significant finding, conclusion, strength and limitation. This should be from your perspective. And is it how it is relevant to your study? I would say make a table like this. Okay, read the paper, take the hints, fill up the table. Okay, this is very much important to see what's happening in the field. Am I working in a actually potential research question? Is my workflow relevant? Many questions can be answered from the literature review matrix. Okay. Do you remember these three experiments? I would say to solve DNA as a genetic material, they took around 30 to 45 years to prove it. If they had a research matrix, review matrix, it's very clear DNA is a genetic material. If you had a review matrix, I will also show you with a real-time example. Okay. So what are the content of review matrix? It should be specifically designed. The paper you are selecting to read and take notes, fill the review matrix should be very specifically aligned to your research question or hypothesis. Okay. So whatever is you have to include in the review matrix, it's up to you. Include that, start reading. I want to give a live example. I was teaching this review matrix strategy in a class where last year, in the Kerala and in freshwater bodies, this brain eating amoeba is causing a big havoc. Okay. But one student came up with an interesting observation. He said that so brain eating amoeba, the case is projected in Kerala 3 years back. They have predicted that it is going to prove. Then he showed me this review matrix. This is so amazing. You see, this review matrix clearly predicted. This is in 2023, where they clearly predicted by 24 that is going to increase. Not only from life science paper, even climate modeling papers, many papers have predicted. This is the beauty of review matrix. Individual authors, Yoder et al., Cope et al., Jamerson et al., they may have missed it. But when you take it in, when you analyze it comprehensively, you can see what other authors have missed. It's very much possible. That's the beauty of review matrix. Okay. So what the second step or advanced stage of review matrix is a meta-analysis actually. A review article or it's just a qualitative assessment of published report, whereas meta-analysis is a quantitative analysis. I will give an example. This is one of my paper where I never sampled copypod or literally extract the DNA of copypod to study the bacteriobiome of copypod, but I collected the bacteriobiome data, metagenomic data published by various authors to answer this one particular question. Is copypod associated bacteriobiome species specific? So we hypothesized that yes, they are species specific. By we end up proving that it's an alternative hypothesis, not what we predicted. So we collected all the metagenome data. Only 11 files were available, more than 12 TB size. Okay, we collected that, and we went for we count. Individual authors of 11 papers may had nearly 30 authors, but they never have done this analysis compiling all their data. But we did. What we found that is Kobe board have a core bacteriobiome and associated bacteriobiome. Okay, so every species have their own core bacteriobiome. You can see that, and here is the bacterial phyla, and the other species have associated bacteriobiome. Okay, this is a very beautiful finding we got. We proved our hypothesis to be wrong, and also why this paper is significant is they also proved that whatever biochemical modeling they have done so far should be recategorized, because these associated microbiome have a biochemical potential. They can modulate nitrification cycle, carbon cycle, many things in the ocean. Okay, so that's how you need to do the meta-analysis. Let's before we proceed, let's understand this research gate. When I told you if you can't find a full or text article, you can also join, create a group profile here, and you can find the actual authors who can share the papers directly. Okay, and building a research network is not a optional; it's a mandate thing during your MSc and PhD. It is a research network that is going to amplify your research efforts. All right, so this platform is ideal platform for that. All right, let's continue to see. So we started with identifying a research domain. We used Google Scholar to read the current article to understand a research question. From there, we came to a hypothesis. Then we design a workflow. Before designing a workflow, how can you use ResearchGate and other stuff is create a concept note. Now, what is this concept note? Here is a research question, not yet answered. My students are still working it. I can show this question. I'm very sure if at all something is going to be solved, my student will do it because they had a head start. Okay. How to increase the stability of drug-loaded bacterial extracellular vesicles for effective delivery, drug delivery, and storage. Okay. So vesicles are extracellular component from bacteria that can selectively fuse with other bacteria. They can carry drugs. The only problem is the storage. They have very short shelf life. So my student came up with a concept note. So what is this concept? Concept note is a one-page write-up. Okay, showing introducing the problem, what is bacterial extracellular vesicle, how it is it, with the proof, citation, and other stuff. Then how they are classified, why one should increase the shelf life, because it's a billion-dollar industry, because you can load a drug to a vesicle and deliver it specifically to particular bacteria. Okay, so with the citation, how many authors have cited that shelf life and needs to be improved for to use the vesicle as a drug delivery system. So this research question, how can we enhance the stability of drug-loaded bacterial extracellular vesicle for efficient delivery? A long-term storage is valid. With the citation, you should prove. Okay, you can also have references to support that. Okay, once you have a concept note, one page write up, now you can find similar experts in the field. You can communicate with them. You can say that this is a research question I want to work. Is it worthwhile to pursue? Can I write my methodology? Not I'm saying this is a foolproof method, but I'm very sure getting guidance, asking multiple authors will set your strategy that you are pursuing the right path. Okay. Uh, did I ask to everyone before I formulate my hypothesis? No, because I have read enough to understand this is something new I'm pursuing. So once you have a hypothesis framed, and then once you have done the literature review and prepared a literature review matrix, I'm very sure you have read a multiple protocols, and now you will know what is the workflow you are going to adapt and how are you going to design your work. So far we have seen how to pen down your idea, gather what domain to work on, how to identify research gap, convert into research question, hypothesis, literature survey matrix. Then comes your concept note synthesis. So right now I am finding this Gen Z especially using ChatGPT and formulating and thinking that you will end up getting a research question. No, ChatGPT works by the data already they have. They cannot create something new. If you ask a current research question to ChatGPT, it will tell you how to find an answer. It will never give an answer. Okay. So one student challenged me that he found a research question that there is no molecular studies, proving how the curcumin interact with the thyrokinase enzyme. Okay, so he prepared a concept note and showed it. I just used these three words, went to a Google Scholar, selected the current year, or just I want to see in 2016 itself, they have clearly proved in this journal that curcumin is interacting, how the interaction happened with the thyrokinase. They have a clear cut proof. Okay, so if you are using some AI to ask question, they may hallucinate and give you many things, which is not at all true. Okay, so a concept note basically should have a title, background of the study, in-depth analysis with the citation, and finally why this question is worthwhile should end with the question. And whatever citation you have included in there, it should be listed as a in the reference section. Okay, so that an expert can look into a concept note and see, or even whether the expert can say it is a good one or not, or even for many small state government run scientific research proposals or many other domains, you can submit your concept note and get it funded. Okay, there are many agencies who are willing to fund your new data, especially during your PhD. You can have your own funds happening if you have this. Okay, so don't be in rush to finalize your question. I would say this is a preliminary question. Again, I copied this question and went into Google Scholar. I found one paper which used lyophilization technique to increase the shelf life, but it is not very successful paper. So in that case, what I will do, see, I have converted the research question into review of literature. Now, before starting my experiment, before starting a wet lab, or before pursuing this research question, I want to write a literature review because now I can gain more knowledges in this field. So this is a question, how to enhance the stability. See the subtitles here in the left-hand side. I have made a subtitles and I'm going to fill these subtitles with the hints I have taken inside. And this is going to help me in framing a literature review. I would suggest before pursuing, try to write a literature review. It's okay. Study the articles, try to formulate a skeleton from your research question, try to write a research review article. This should be, in my perception, this should be done before first year of your MSc if you are finding an MSc project. If it is a PhD, before first year of the PhD, you should have a literature review published in your domain. Okay. So again, went through this paper. It's not a worthwhile paper to pursue because that method is not foolproof. So they did not increase the shelf life. So another student used AI to see a research question. She wrote a concept note using AI. Okay, and she said that there is no study tested tulsi extract in Ajinomoto, monosodium glutamate effect on Drosophila, how they are killing the neuromotor function and other stuff. Again, we went to Google Scholar. We found multiple papers that have already used. So the claim that no study has tested, that's why I want to work, is not a valid claim. If don't choose a topic because no one has done it. What is the you are going to invest your time and energy? What is the outcome? How it is going to be useful to the society? No way a PG student know what is useful for the society. So they will never ever create a question or a project which has a social impact. How to have a or how to create a project that has a social impact? Understand what's happening in the research domain. Read article. That's no other way. Spending more time reading article, then getting your head around and see what's happening in the research field will actually give you a profound research question which you may end up having a social impact. Or if you're hallucinating like this, no study has done, already there are tablets in commercial market from tulsi extract, regulating various other things. All right. Even when you submit a proposal from ChatGPT, if I copy a content and ask ChatGPT, did you write this? It will say yes, I wrote this. Okay. So do not use ChatGPT to ask a question. Okay. Or You can use ChatGPT like this. A student want to understand a molecular mechanism of HIV against human immune system. He asked ChatGPT to derive a 5-unit syllabus with the content in this topic, and it has given a reference books and other stuff also. You may use ChatGPT. You can understand each and every vocabulary to get the expertise in the particular domain before asking a research question. Or you can collect the research gap yet to be answered from various articles, consolidate them, ask ChatGPT to ask a research. These are the research questions in homologous recombination. Right now in 2025, that is yet to be answered. These questions have never been answered. This is unanswered questions. Likewise, this is consolidated from ChatGPT, but the feed is given from the text taken from the research gaps from various articles pertaining to homologous recombination. Okay, so all right. So there is another study that actually, have a research question. They converted a research question into a review article structure before, deriving an experimental protocol. You can do this. Not necessarily every researcher has to do this. Even for a people who seeking job, for example, this is a job role described by Biocon. All right. These are the individual roles described by a life science industry. So a student have taken a particular job role, understand the questions, understand what each and every question is. He converted the job role into problem statement. Why they created such a role in that industry, especially in this endotoxin and bioburden testing site. What is their requirement? And based on their role, he converted into a problem statement. That problem statement is now converted into a structure of a review article. If you are someone looking for a MSc, after MSc, if you are someone looking for a job, do not waste your time in pursuing research that means nothing to you after your graduation. Okay. Better select a particular role in a company, understand their job responsibility, convert it into problem statement, try to draft a review outline from the problem statement, try to publish a review in MSc first year, do your project in the same domain. When you sit for an interview, I guarantee you will get a job. Here is a proof. Okay. These two students, they are very clear what they want to do after their MSc. That's how they selected their job role, converted it into problem statement. They published a review. They didn't even publish their actual wet data. Okay. They don't have a time. And this student, he just gave one interview after that, sitting in a food tech industry with, a review article in the food tech and thesis that is already generated data, what they are already doing in the industry. That's what the thesis is all about. They didn't even ask much question. They just gave an offer letter. Okay. This is how you need to orient yourself for a specific job. Industries are constantly complaining about lack of skilled manpower. Everyone has a degree, no one has a skill. It's like our academic institutes are producing a mass number of educated illiterates. That's how the companies are feeling. They are constantly complaining about lack of skills. This is how you need to use your 4 semesters in masters to orient yourself towards particular skill. Okay. So again, yeah, one student came up with some bizarre, a question to study gut Earth microbiome as a vessel for horizontal gene transfer of plastic degrading genes. So there are mechanism of horizontal gene transfer among bacteria, which is plastic degrading. Why such a specificity? I dig up into it. Suddenly I saw, one search in the Google Scholar gives a paper that is already, similarly, they have studied the horizontal gene transfer in antibiotic resistant genes. You know, bioplastic degradation, when you went into the paper, you can see they are already done. But in the last paragraph of the discussion, they were talking about what is yet to be done. Okay. See, apply this if an ideal hypothesis have these 3 words inside, if, then, and because. You apply it here. If antibiotic resistant genes can be, interactively disseminated in soil earthworm system as a rapid egestion capacity of earthworms, then PVC microplastic may exacerbate the spread of antibiotic resistant genes in the system, because they not only act as because, the third word. Okay, so ideal hypothesis should have these three words: if, then, because. So to test it, now the authors have used this experimental workflow to test that particular hypothesis, and they did a decent job. But in the last paragraph discussion, they have many unanswered questions they have discussed. So if at all you are looking for a research question in this domain, these are the unanswered research question. From the last paragraph, every wet lab research article will have this: what is yet to be done. That's where you have to collect the questions. Now you check each and you treat each and every question as a preliminary question. Again, check it in the literature review. See that these questions are unanswered. And also you ask your question. If I'm going to spend 2 years in answering this question, okay, is it worthwhile? Is it contributing significantly to the society or human well-being? If not, it's not worth pursuing. Okay. CRISPR-Cas, now we are using it to engineer human babies. Actually, it's a basic science research. Virologists and bacteriologists, they are understanding bacterial immune system, how they are actually, reflecting or protecting a bacteria against a incoming viral genome, which end up in the application. So all basic research will also have a impact impactful application. You should know this. So this study can also be improved in my perception. This study did not assess diversity of bacteria directly from the microplastic surface, because there are many studies that have collected microplastic and assessed their eDNA, and we have also shown that the studies have shown that eDNA have a better absorption to microplastics. So you can simply collect those DNA, and it is very common in marine system and other aquatic system, and see how this works. So these are the lacunae in that domain you can address to, instead of clinging on to the basic questions. Okay. So literature review matrix, how the DNA has been proved. So if you end up publishing such a article, definitely your article will get more citation. So what is this citation? Okay. If your articles are not going to get, otherwise of saying, if you don't have 1,000 citation for your published work, you cannot even apply to IITs and IIC. Okay. The reason IIC, IITs observe people only from, who got post graduation or undergraduation from outside India, or PhDs from outside India, is usually their publication have more citations. That's the reason. It's not, they are not discriminating against the geography. Actually, they require cited published article in your profile. They require well proven academician, because the institutes, IIC and IITs, are going to invest on you on, in huge way. People monies are going to invested on you. So they want to see that you have a proven track. Okay. So what is this citation? Let's understand what is this citation. I will take this professor's from IIC. I don't know him personally, but we'll use him as an example to understand what is this citation, H index, item index. So if you don't have, I said, 1,000, 2,000 citations, you can't even apply for scientist or other position in leading research institute, for which you need to publish a article. You need to do a research which is worth publishing. If you are keep on publishing, if your article is not cited, you are actually going to degrade your career. Yes, it will damage your career. If number of articles are high, if number of publications are keep on increasing and the citations are not getting up, you are actually a burden to any institute. You are not institute and not to yourself also. I will tell you how. You see a Google Scholar profile of this professor, okay, with total number of 178 citations. He was publishing since 1970. Okay, his H index is 52, meaning he has overall 21,000 citations, but his H index is 52, meaning at least 52 papers in his profile have 178 citations. So H index is the largest number of H, that H article have at least H citation each. So what is happening here is they are just considering number of citation, number of publication, and they are equating. Say for example, among 178 citations, 178 article he has published, only 52 articles have 178 citation. That's why their H index is 52. Then what is H10 index? Among the all article published, at least 10 citation. How many articles have 10 citation? It is 104 articles. Okay, so this is the H index and I-10 index used to assess the quality of the research work you publish. Imagine you are keep on increasing your number of publication and nothing is getting cited, meaning your H index and I-10 index will be one or two. You may have, 1,000 articles published, but if your I-10 index is not even one, what is the use of publication? So institute will see H index along with number of citations. So here, pause the video and tell me what is the citation of this author who has published 7 papers, 50, 45, 30 citation for third paper, 25, 6 paper, 10 citation, and seventh paper, 5 citation. What is the H index? H index is 6. Why? At least 6 papers have 7 citation. Number of publication is 7. So at least 6 papers have 7 citations. So the H index is 6. Okay. That's how the H index is calculated. Imagine, remember, you need to have a citable publication. You need to have a, for citable publication, you should have answered an unanswered question in the current domain. To have an unanswered question, you need to have a right workflow. You need to have a literature review. You need to have a hypothesis, which is derived from a potential research question, confirmatory research question. You may have a 10 to 15 preliminary question, confirmatory question. From there, you need to derive. That's all these presentations are. I just want to introduce whenever you are writing an article, if you are using a Zotero or other citation formatter, you can insert citations easily and the references in API style. Otherwise, always use this cross-site website where you can simply copy and paste your DOI and you can get your, just pasting the DOI, you can get your entire article in API format. If you want, you can make, this is cross-site.citation.com. You can put it in the Google, you can get to this website easily. All right. So enhancing the stability, like I said, this is a concept note. From there, you can have a research article derived. Once you start writing a research article, you can have a skeletal of the article, individual things you need to include it. Finally, you can get into a research article. Usually, the journal will accept plagiarism. You cannot simply copy and paste here. You need to use your own words. Should be less than 10%. Okay. I will give you a classic example of how ChatGPT hallucinate here. Okay, a student have copied all his references. He gave to ChatGPT, asked ChatGPT to write it into APA format instead of using a thing. So ChatGPT gave a very good APA format. It is very evident that it is from ChatGPT. This asterisk denotes that. When I click this DOI number, it does not exist. When I click on another DOI number, see the title is Nanostructure Lipid Carrier Enriched. But here, see the title, it is completely different paper. This is called hallucination. AI will hallucinate. When I click this DOI number, see the article Advances in Hydrolipid. You see, it is going to some different domain, completely unrelated. This is hallucination. They just gave some divine number, okay, instead of converting them. This is the danger of ChatGPT. When you communicate it, this is considered as a plagiarism, and you will be barred from many things. Okay, so while using ChatGPT, please be very careful. All right. So after inserting, I will tell you a quick hint on how to find a research journal for communication. Once you are done with your review article, insert line number, other stuff, go to a journal finder. In a Google Scholar, just type journal finder. You will have many journals from Elsevier, Spingeling, Wiley, Taylor, and Francis. You can simply copy your abstract and paste it in the journal finder. It can give you an array of journals where you can communicate. You can even see whether the journal demands article processing charges. If your project or you cannot self fund it, you can simply choose a closed journal. You can communicate your article where readers will pay, and you can select all those things. What is that reply time, acceptance rate, everything you can read it here. Okay, so I would say for BSc or MSc or even PhD, higher studies, job, or a startup. For all three options, you need to know how to read and understand a research article. So if you are told you need you want to go for a PhD from a MSc, better choose a domain today itself. Okay, and pinpointly focus on where you want to go, which guide you want to select during your MSc, which country, which institute, what is their research domain, better choose a research question from their current publication and start working. Okay, this Google form and questions I have created to make you more focus on what you want to do. So here, once you selected a job, higher studies, or startup, I have oriented a research following research question, how to check or how to select a particular field. Whatever you are selecting, job, startup, or even higher studies, having a review literature in first year and orienting your entire wet lab towards that in the second year will definitely get you what you want. Okay. So these are the questions if you answer for a job. Even for a job, do not say I want to go to biocon. Biocon is there are more than 28 departments. Even various biocons are there. Biocon biologicals, biocon therapeutics, biosphere. Which biocon? What department? They have hundreds of departments. Even if you select one biocon biosimilar, there are hundreds of departments. Which department? What is your job role? Which country? What is a particular job role? If this is a job role, they have clearly declared. From here, you can clearly convert this responsibility into problem statement. From there, you can derive a literature review. You can design your entire wet lab in understanding that. You can go to Biocon and tell them that I have a review article. I want to design a wet lab in this. Can I take an internship in your company? If there is a feasibility of 6 months project, better do a 6 month project. That I have seen many students who spend a 6 months project in their target industry will get naturally absorbed into the industry. Okay, so job or higher studies, that's the way to specifically select what you want and progress your career. So like I said, the last slide, first, gain the knowledge in your selected domain, find a research gap, convert a research gap into research question, design a set of hypotheses, which will lead you to list of experiments, perform the experiment, select the journal, communicate. So that's a way, that's a strategy to do a quality research work, which is worth publication. All right. So I will also introduce a little bit of barcode biotechnology. We are basically a sequencing company, DNA sequencing company. And recently we are into various academic activity like organizing project works also. Usually after DNA sequencing, customers ask us how to analyze the data, how to build a phylogram, how to submit the data, how to edit the chromatograms. So we thought we will come up with a comprehensive program where we are actually training UGPG students and even PhD students in DNA sequence analysis. This time we are offering 2 DNA sequencing reaction for free under this program, 21 days mini project program, which is going to start by October 7th this year. All right. So this program will give you, you can send the samples actually to Barcode Biotechnology. They will analyze the sample, get the sequences. They will give the sequences. Using your own DNA sequences, you can actually perform the DNA sequence analysis. This is a online program where you will send the sample to the company, get the results and do the analysis. We will guide you in preparing a thesis work for if you are doing a mini project in your 5th semester or project before your actual project. This thesis work we will take care. We will guide you from A to Z. If you are choosing something new species, a quality data, we will also guide you in manuscript preparation and communication. If you want to check our previous manuscripts and other data publications, here is a link tree slash barcode biotechnology. You can check the list of customers who has published our publication. I hope this webinar is very much useful to you. For certifications for attending this webinar, I will be sharing a link in your WhatsApp group and also a Excel file which will contain your name and the certificate number corresponding to your name, which you have to use to download your certificate through the link we are going to post in your WhatsApp group. So link and the Excel file, do look in the WhatsApp group. Hope this webinar is very much useful. If you are interested in registering for this program, please free to scan this QR. There is a small project fee that's around 5,550, but I'm very sure this is more than what are you going to take, because to sequence do 2 DNA sample, it will cost around 8,000 in the current market. So you can select a plant, animal, or microorganism, anything you want. We will sequence it 2 samples or 1 sample, 2 genes, both are possible. Do 2 DNA sequencing reactions will be sponsored here. and using your own sample, you can run the entire analysis. So thank you for this webinar. Do check your WhatsApp group for the further details. Thank you.